Hello everyone, my name is Austin Weissong, and today for our microbiome course I will be presenting on a paper titled Tumor Microbiome Links Cellular Programs and Immunity in Pancreatic Cancer. So pancreatic cancer, why should we care about it? The most common form of pancreatic cancer is the focus of this study, and it's pancreatic ductal adenocarcinomas. These are very aggressive. It's a very aggressive form of cancer and has a very poor prognosis. So this paper wanted to establish whether there was a connection between microbial host associations and prognosis. So it had been demonstrated in research prior to this that there was a microbial component inside the tumor environment, that there was bacteria in there. And this paper actually provided quite a bit of evidence that would lead them to determine that there is a strong correlation between particular bacterial host associations and cancer behavior, cell behavior, and consequentially the prognosis for the patient, the immunological impact. So common abbreviations that I'm going to use throughout this PowerPoint are PDA, which is pancreatic ductal adenocarcinomas, SOME, which is single cell analysis of host microbiome interactions, and essentially I'm going to talk about this a lot more later, but it's a software method tool that the authors use. scRNA, which is single cell RNA, and generally the SC prefix might be on the front of some other words like DNA, scDNA, so just know that SC generally just means single cell. GI is gastrointestinal. DEGs are differently expressed genes, and I'll be touching on that a little bit later when I focus on bacterial associations. IMERs, or IMER, is the infection microenvironment reaction, and similarly TMERs are tumor microenvironment reactions. So a little bit of a background on pancreatic cancer. The pancreas is an organ. It sits kind of in between the back of the stomach and the intestine. So it helps the body manage its blood sugar. It secretes hormones and also enzymes for digestion. And pancreatic ductal adenocarcinomas, they actually start in the pancreatic ducts. And that's they're used to carry enzymes out of the pancreas into the gastrointestinal tract. But that's kind of where the problem with this form of cancer lies, is that it's kind of in between everything. It's in the middle of the body cavity. It's sandwiched in between a bunch of organs. So when when this form of cancer begins to arise, there isn't really a very good way of detecting it. There's no way to feel for it like you would for, say, breast cancer. and there's no no, well, until it becomes advanced, it's very hard to see it with an X-ray or an MRI because of the fact that it is in between several organs. It's kind of obfuscated. Um, and its initial symptoms are very general. Jaundice, fatigue, loss of appetite. They don't really point towards a definitive differential diagnosis. So again, it's very easy for this cancer to get missed until it becomes a late stage cancer. So risk factors, there's general ones that typically increase the risk for most forms of cancer: smoking, excessive drinking, chronic inflammation, and obesity. All of those typically will increase the risk of cancer for most cancers, but familial presence of this cancer, if you have a relative that's had this cancer, then that's generally a pretty big risk factor. Type 2 diabetes, which is kind of linked to obesity, and advanced age as well, all are risk factors. And genetic testing is actually a major component of monitoring for this this cancer and someone's risk for it. And there's certain genes that will increase the risk. I think the BRAA gene is one of those. So the necessity for novel approaches when it comes to PDA research and treatment, due to how aggressive the cancer is, how bad the prognosis is, it's going to come down to palliative care after it's diagnosed. Typically gets caught somewhere in the stage 3, stage 4 range when it is likely already metastasized. So the push for treatment for remission is not really there a lot of the time. It's become delocalized. So a lot of the treatment includes alleviating pain at the moment. But developing new strategies to personalize this is very important. A lot of the advances that have been made in cancer treatment recently in the last 20 years have been focused on personalizing treatment for patients based on their form of cancer, their specific cancer. So that doesn't really, there's not been much personalization for pancreatic ductal adenocarcinomas. So finding new factors that might inform a treatment strategy, inform prognosis, let people know how much longer they may have, or generally anything is welcome due to the lack of anything at the moment. And the predictive models that are used at the end of this paper they could be very useful in the future once there's more research into this topic off the back of this study, based on someone's specific cancer, someone's specific makeup, the composition of their microenvironment may be able to inform in the future what needs to happen in terms of treatment, in terms of the prognosis. So the aim of this study generally is to piggyback off of some research that had already been done into the topic. Pancreatic cancer tumors, it had been determined that there was a bacterial component to their microenvironment, and and generally investigate that deeper. The problem with the studies before this one had been that there's a lot of fibrotic tissue inside of this specific tumor, which made it hard to disentangle and determine the relationship between the host tumor cell and the microbial community. But using the SAMI pipeline, the authors were able to disentangle those relationships and to investigate what bacteria are present, how the microbial tumor associations impact the cancer behavior, such as like motility, and what the immunological impact and survival rates have to do with this makeup and these associations. So the first bit of methodology is going to be the Novosec platform, which is the sequencing platform they chose for this investigation. And they were focused on analyzing 16S rRNA, specifically the amplicons, to generate a metagenomic sort of picture. So they used Novosec 6000, which it's a form of Illumina sequencing. I think we covered it in week 2, but essentially it uses flow cells and nanowells, fluorescent dyes, all of that to sequence. It sequences quite a bit. It's very good when it comes to throughput, and it's very good at reading shorter sort of reads, prokaryotic ones. I would say that it is very well tailored to this investigation, as they're not mapping a eukaryotic genome, and they're not even mapping in great detail each individual taxa. They're identifying the taxa, but they're not looking for specific mutations or single nucleotide polymorphisms or anything like that. So I would say they made a very good decision when it comes to the Novosec platform. So the SAMI methodology, it's as I mentioned before, it's kind of like a all-encompassing software pipeline thing that they funnel the data that they generated using the sequencing. They had 2 cohorts that they sequenced, and it was I think 44 different patients, different samples, and they funneled that all into SAMI, which is uses filters to kind of get rid of the bad data, and it uses K-mer libraries. It cross-references libraries to determine what taxa are present, and then it uses a series of tests, a correlation test against negative controls, and comparisons, essentially just to weed out anything that shouldn't be there based on data sets that it had been trained with. And that at the end of the pipeline leaves the denoised microbial profiles, and then more importantly, it leaves them with their paired host cell, the tumor cell that they were sort of associated with. So the microbial makeup of a single cell essentially can be dived into using this method, this denoising platform, which is why it's so important, and it essentially makes this entire study possible. quickly, this is just a little visualization I wanted to put in here that they provided for the SAWME pipeline, starting with the sequencing, going through Kraken to Unique, which is their K-mer taxonomic profiler, and then the two tests, the KCT test and the CLQT test, that weed out anything that really doesn't belong or isn't relevant, false positives. So the statistical analysis used, they generally used 3 different tests or formulas to generate their figures to interpret the data that they got after they put everything through SAWME. The first one was Wilcoxon tests, which essentially just allowed them to interpret the the two cohorts, independent variables of the 2 cohorts, and just apply them. Finding the shared value was really important. Shannon Diversity Index, another statistical method typically used to determine the ecological makeup, and they applied it to determine the taxonomic makeup of the association between the microbial community and each individual tumor cell. They use that eventually for cancer cell properties and what is affected by the makeup of the community. And finally, Fisher's test. They use it to develop contingency tables, and it's a pretty simple equation and formula. It's to build these contingency tables to disprove the null hypothesis. Essentially, they bring it up quite a bit when they're talking about their predictive models that they make after evaluating all the data. So to prove that the predictive models are useful. And this is just a graphical abstract. Feel free to pause on this if you want to see essentially the methodology that I've already gone over a little bit. So this is the first part of the results, and it's taxa present in pancreatic ductal adenocarcinomas. So the taxa that they found were GI taxa, gastrointestinal taxa. Some of the species were Campylobacter concisus, Clostridium difficile, Fusiobacterium nucleatum, and a couple of others. And the presence of this was at a much higher level than any other organ-associated community of organisms. So that initially just informed the researchers about what kind of bacteria they were really dealing with, and it was gastrointestinal bacteria, which makes sense because the gastrointestinal tract directly contacts the pancreas. The ducts connect to the gastrointestinal tract, and the general proximity of everything makes sense. So essentially the bacteria traveling into the tumor is what made up the majority of the microenvironment. Um, so this figure really um, it's about testing the SAMI pipeline, and more interestingly is the GI overlap. Again, it kind of started to paint a picture when it came to the actual character of the tumor and the tumor's microenvironment. And that really is that. So SAMI, it was correctly isolating things that had been found in previous adenocarcinoma studies, or when it came to the pancreas research, it was correctly denoising the other taxa human things and general junk that was in the data set, which is important, but more importantly is that the only set of data that was also compared with in this test that had any matches was H. pylori and Salmonella enterica studies, sequences, and again, those are gastrointestinal organisms, and it started to become very clear that this was a gastrointestinal association within the tumor. So tissue types in the figure 2D, they kind of go into how different bacteria generally just associate with certain cell types. And that's why if you look at the figure, in both cohorts, the SCPDA1 and SCPDA2, the bacteria association within the tumors are clumped and consistent. This is pretty important because it generally paints a picture that the bacteria associated cells are grouping together and are forming a mass. They are all in contact, and gene expression and other things signaling are probably conserved within that area. And again, this kind of informs the prognosis just a little bit, knowing that these associations are forming within the tumor, which otherwise doesn't really have an evolutionary reason to have an association like another organ, like the gastrointestinal tract would with its associations. So this was a pretty interesting discovery. So more on the microbial tumor associations. It was determined that based on the data sets, the tumors had the largest number of associated bacteria per cell, which again is a little bit concerning. There isn't really an evolutionary background to this. So, and obviously infections of bacteria typically provoke an immune response. So they touch upon that a little bit later, but the bacterial association between tumors and their host cell association thing really stands out. Nothing even really comes close to it in the rest of the counts and studies in the two cohorts. So there's a significant association, likely significant changes to the phenotypic presentation of the cancer due to signaling, due to the presence of these bacterial components. So that leads into the enrichment. So the last three figures have really all led to this. As you can kind of see in the figure, the enrichment in presence and character is by far the strongest between the tumor and all of the rest of the bacteria that were observed within this environment, with very weak enrichments and associations between other cells, endothelial cells, fibroblasts, myeloids, and again, GI bacteria having very strong enrichments. The immune cells were found to be co-localized with the bacteria. But even though these immune cells are present in the tumor and are present amongst all these bacteria that have actually invaded and made their way into the tumor, there were only 3 bacterial immune cell enrichments. So there was very little evidence that the immune cells were having a similar association to the tumor cells as the tumor cells had to the bacteria present in the microenvironment. And that informed possible bacterial tropism towards the malignant tumor cells. So gene expression is the focus of this figure, and it really paints a picture between the gene expression in each cohort and how the bacterial association and co-localization changes the expression of genes within the tumor. It significantly increased expression of EDIL3, which is an integrin ligand, and it's implicated in angiogenesis. It also changed the association of the bacteria, changed expression of CRABP2, which is an oncogene, and it was upregulated in the presence of H. pylori, and specifically another form of CRAB2, it enhances pancreatic cell motility. So this could possibly be a factor not just for something like angiogenesis, but for metastatic development, becoming even more aggressive, metastasizing. And other genes, OLFM4 expression was much higher, as can be seen in the figure, and that is it's a stem cell marker that increases epithelial regeneration. And generally all of these upregulated genes found in these associations had negative prognoses, which just kind of lends itself to showing that the microbial association was not good for the patient. This figure on gene expression is also, it's very dense, but it essentially builds upon the last figure and shows that bacterial association was increasing the activity of genes and pathways that promoted cancer regression. Everything from decreasing neutrophil, extracellular matrices and traps, complement cascades, and immunological hurdles. It just allowed the cancers to overcome it, really. And this is the big one, as I stated. It's the impact on survival rate, and it really is a stark figure, with the pink line in each of the graphs representing the survival probability dropping in patients who had cell-associated bacteria within the tumor. And it clearly shows a significant drop in survival rate when the bacterial associations are present. It's touched on a little bit inside the article that the presence of the bacteria likely was leading the immune system to trigger more and more inflammation in the area of the tumor, which was suppressing the ability to fight the aggressive cancer. So the discussion, again restating that the investigation of the microbiome and pancreatic ductal adenocarcinomas is very important. It probably plays a key role in modulating tumor activity. And based on the results of this study, there should be further investigation to see if there is a treatment pathway that can be informed by this. The one problem, or at least the one downside, is that there wasn't a correlative relationship, or there wasn't a causal relationship. There was a correlative one established between prognosis and the tumor microbiome. So the conclusions, really the takeaways, is that the microbial composition of the PDA tumor microenvironment it's impacted by gastric bacteria. The associations do seem to play a role in changing the cancer presentation, and the presence of the bacteria is associated with very poor prognosis. I would say the strengths of this study really come from the SAW-Me pathway, but some of the areas of improvement, I think I touch upon it in the questions here, is that they probably need to do more tests. 44 samples is significant, or statistically significant, but they could always push for more, generating larger cohorts, and investigating with maybe an even more focused scope than they did in this. So I'm just going to finish it up with my questions. If host microbial associations impact tumor behavior in PDA, could they broadly modulate other types of cancers? What potential treatments, therapies, or surgeries do you think would help reduce these negative associations? Could probiotic supplementation for the GI system regulate and minimize key bacterial species that cross over into PDA tumors? And if you were tasked with continuing this investigation into the impact that the microbiome has on tumors, what details or strategy would you pursue? And finally, do you think that the microbiome will become a bigger component of medical treatment in the future? And if so, in what ways do you see it growing and being used? And finally, here are my references. I hope you enjoyed my presentation.